Hello friends, we have been learning very interesting unit from your paper second, that is plant physiology. The name of unit is plant tissue culture. From this plant tissue culture we have to get idea about the today about callus culture. So what is callus? Then which methodologies are used in the preparation of a callus, and what are the we have to say that application significance of this callus culture. Friends, callus is a undifferentiated, very interesting point you have to keep in mind. Callus is a undifferentiated, unorganized mass of a plant cells. This is a undifferentiated, unorganized mass of a plant cell. This definition you will have to always remember. Undifferentiated and unorganized mass of a plant cell is known as a callus. It is basically a tumor tissue. It is basically to which usually forms on a wound. We know that whenever any injuries made to the plant, at that time some swellings we can find there, some tumors we can find there, and that tumor swellings are nothing but the callus, which is unorganized mass of a tissue is there, my friends. So, callus cells are parenchymatous. Callus cells are parenchymatous. My friends, we have got idea about the callus. Callus is a undifferentiated and unorganized mass of a plant cell. It is basically a tumor tissue which usually forms on a wounds of a differentiated tissue or the organs. Callus cells are parenchymatous in nature, although not truly homogeneous. Keep in mind, they are not homogeneous, they are parenchymatous. On examination, callus is found to contain the same quantity of a differentiated tissue besides the bulk of a non-differentiated tissues, isn't it? Differentiated and non-differentiated, same quantity we can find there. So we have to say that the callus, again I have to tell that callus is nothing but the unorganized and undifferentiated mass of a tissue is there. Callus formation in vivo, we have to say that, is frequently observed as a result of the wounds at the cut edges of the stems and roots of the plants. In the nature, I have to say that callus formation in vivo means in nature, isn't it? In vivo means in nature, and in vitro in the sense in the test tubes. So here in vivo, in the nature we can find there is a callus formation, isn't it? And that callus formation is possible to see on a wounds at the cut edges of the stems and roots. Invasion of a microorganisms or damaged by the insect feeding usually occurs through the callus. An outline of the technique used for the callus culture and initiation of the suspension culture in given in the next diagram that we have to learn. My friends, you have to keep in mind when and where we visit to the any garden, any field, any we have to say that the forest, at that time many trees are there, many plants are there. On that stem we can find some tumors are there, and that tumor are nothing but the unorganized undifferentiated mass of a tissue, that is a callus. In vivo we can find these things. So that is that we have learned here right now. My friends, this is the simple, I have to say that, the diagrammatic representation here of a shoot tip meristem we have taken cultured and a micro propagation. So here what happens actually, which things are going to happen, and where the actually suppose explant plant is there, from that plant we have taken the shoot tip. In the sense, why shoot tip taken? Meristems are there. Meristems means the cells having capacity to undergo division and redivision, very easy. Culture medium, on this primary culture medium we have added the explant is there, sorry, we have added yes, explant is there definitely. Then shoot development with primary axillary shoot, then root and shoot development is there, and root plantlet, and this finally we have to plant that plant into the potted clay pot, isn't it? Clay pot we have to plant the plant itself. So this is actually micro propagation, simple method of a production of a we have to say that, or development of a plant. So micro propagation, this technique also you have to keep in mind, because this technique playing important role at the time of learning this total topic. Here, why I have shown that, first of all, explain diagram of micro propagation, and right now we have to get idea about how the callus culture is developed, isn't it? Here, my friends, you have to keep in mind keep in mind, surface sterilized explant, first of all we have to select surface sterilized explant, we have to select, isn't it? Then that surface sterilized explant is to be this one, surface sterilized explant. I have made it little bit pink in color or red in color and put on the agar medium, isn't it? And after incubation we will find the callus is developed, undifferentiated mass of a tissue. So we have added a small amount of explant is this one on culture medium, and later on after incubation we will find this type of a, this much size of a undifferentiated and unorganized mass of tissue, and this is the callus. This is the callus. Now subculturing is there. Subculturing in the sense this callus culture, callus is there, this callus is again taken into a liquid medium, isn't it? And we will take into liquid medium, and then what happens actually after incubation we will find that is a suspension culture. Callus culture is converted into a suspension culture. What is use of all these things, that we have to get idea. Now my friends, you have to keep in only your mind, in case of callus culture only you have to take the explant, sterilize that one, and that sterilized explant is to be put into the or on the culture medium, incubate that one. After incubation you will find there is a increase in the size of that explant, or that is because of division, redivision, unorganized and undifferentiated mass of tissue develops after 2-3 weeks, isn't it? And that is nothing but the callus, and method is nothing but the callus culture method. Now that callus again we have taken into, transfer into the liquid medium, and that callus liquid medium kept on a shaker, and after 1-2 hours we will find there is a suspension culture. Cells get separated from that group, isn't it? Now explant for callus. Explant means starting material for the callus culture may be differentiated टिश्यू फ्रॉम एनी पार्ट ऑफ द प्लांट. कुठलं एक्सप्लांट वापरू शकतो कॅलस कल्चरसाठी? शूट वापरू शकतो, इजंट इट? शूट टिप वापरू शकतो, रूट टिप वापरू शकतो, स्टेम टिप वापरू शकतो, लीपचा काही पार्ट वापरू शकतो, फ्लावरचा स्मॉल पार्ट दॅट वी कॅन यूज. द सिलेक्टेड एक्सप्लांट टिश्यू मे बी डिफरंट स्टेजेस ऑफ ए सेल डिव्हिजन. ते आपण जेव्हा सिलेक्ट करतो एक्सप्लांट तेव्हा ते वेगवेगळ्या स्टेजेसला असतं आहे, इजंट इट? सेल डिव्हिजनच्या, cell proliferation and organization into a different distinct specialized structures. If the explant used possesses a meristematic tissue, keep in mind, जर आपण एक्सप्लांट जे निवडलेलं आहे कॅलस कल्चरसाठी, जर त्याच्यामध्ये मेरिस्टेमॅटिक सेल्स असतील तर काय होतं की देन कॅलस डेव्हलपमेंट प्रोसिजर गेट स्पीड, rapid rate of development of a callus, we have to say that increases for that one. So here you have to keep in mind, at the time of callus culture methodology, at the time of selection of a explant, you will have to see that meristematic cells are present into the explant. That part of the root, that part of the stem you have to select, isn't it? Means shoot tip or root tip, where the meristems are there, where the meristematic tissues are there, that part you have to select and put on the culture medium in the test tube, incubate that one. Definitely immediately you will be able to get the callus culture from that one. So for the cell division, meristematic cells are there, they are originally having capacity to undergo the division and redivision fast. That process can take place, and definitely the callus will be developed very very soon. Means my friends, here a question may arise in your mind that why not take the leaf material? Leaf, each and every cell having a totipotency, means capacity to undergo division and redivision, although that cell is not part of the meristem, my friends. So that also we can take, but for the speed up things we have to select this, that is you have to keep in mind. Now suspension culture from the callus culture, that already I have said to you what things are there. Suspension culture can be initiated, that is possible by transferring friable callus to the liquid nutrient medium, isn't it? Whenever the callus is produced, that callus we have to transfer to the liquid nutrient medium containing container. As the medium is liquid in nature, the pieces of callus remain submerged. Pieces of a callus remain submerged. This creates anaerobic condition and ultimately the cells may die. For this reason, the suspension culture have to be agitated by rotary shaker. Due to agitation, the cells gets dispersed besides their exposure to the aeration. My friends, सस्पेन्शन कल्चर कसं तयार केलं तर कॅलसपासून केलं. आपण काय केलं, पहिल्यांदा कॅलस तयार केला. कॅलस तयार करण्यासाठी आपण काय घेतलं होतं तर मेरिस्टेमॅटिक सेल्स असलेला प्लांटचा पार्ट घेतला होता, दॅट इज अ एक्सप्लांट. तो आपण कल्चर मीडियामध्ये ठेवला, इन्क्युबेट केला. काही दिवसानंतर आपल्याला कॅलस मिळालं, कॅलस कल्चर मिळालं. मग ते कॅलस कल्चर आपण घेतलं आणि कशामध्ये टाकलं, लिक्विड मीडियममध्ये टाकलं, लिक्विड न्यूट्रिएंट मीडियममध्ये टाकलं. आणि नंतर ते लिक्विड न्यूट्रिएंट मीडियम आपण काय केलं, शेकरवर ठेवलं आणि ते शेक केलं, इजंट इट. आणि त्याच्यामुळे त्यातल्या सेल्स ह्या सेपरेट होत गेल्या. आता समजा आपण तसं न करता, शेकरचा वापर न करता तर कॅलसपासून सेल सस्पेन्शन तयार करायचं, इट इज नॉट पॉसिबल. बिकॉज व्हॉट हॅपन्स ॲक्च्युली व्हेन वी पुट ऑर ट्रान्सफर द callus into the liquid nutrient medium, at that time it gets submerged, and in submerged condition there is an anaerobic condition, no oxygen is available to the cells and cells may can die. Each and every cell, living cell, requires oxygen for their survival, and that's why we have put that the transferred callus culture and nutrient liquid medium container put on the shaker, and shaking has been started. And not only the separation of the cells takes place from the callus, but also the oxygen is made available to the each and every cell because of the agitation. Keep in mind, very interesting. Now, technique of callus culture. During the callus culture formation, there is some degree of differentiation both in morphology and metabolism, resulting in the lose the ability of photosynthesis. It produced when explants are cultured on appropriate solid medium with both the auxin and cytokinins in a correct conditions. 2,4-D are commonly used. It is an unspecialized, unorganized, growing and dividing mass of cells. A callus is a block of tissue. Friable callus shows in loosely associated cells, and callus becomes soft and breaks apart easily. Habituation. It reduces the requirement of auxin and cytokinins by the culture during the long-term culture. Compact callus shows densely aggregated cells. Callus culture may be a compact or friable. So my friends, with this you'll have to keep in mind there are the two types of the callus we can get during the callus culture. One is a friable, another is a hard, means soft and hard. These are the two types of the callus, soft and hard. Soft means friable, hard means very very compact. Now, it can be used to isolate single cell with the stem cells like the properties proteoposensity, no photosynthesis, grows in a dark, can be maintained indefinitely. There are three stages of the callus culture. You have to keep in mind induction. Cells in the explant differentiate and begin to divide. My friends, as we know that in case of callus development, the callus proceeds, means formation of a callus passes through the three stages. First one is induction, another is proliferative stage, second one is a morphogenesis stages. These three stages are there: induction, proliferative stage and morphogenesis stage. In induction stage, cells in the explant differentiate and begins divide. In proliferative stage, second stage, we can find there is a rapid cell division. Proliferative stage, rapid cell division. And in case of morphoid, differentiation and formation of an organized structure, specifically process that leads to the plant regeneration from the somatic cells. So induction, division, differentiation, organogenesis, somatic embryogenesis, that are the further stages we can find. Now, morphological characteristics of the callus right now we have to study. What is the characteristics of the, how we will be able to see that this is the callus only. Callus tissue proliferate as a amorphous masses of the cells having no regular shape. तुम्हाला कॅलस तयार झालेला जो दिसेल त्याच्यामध्ये कुठंही रेग्युलर शेप नव्हतो, नायदर इन द विवो नॉर इन द व्हिट्रो कंडिशन. म्हणजे जरी स्वतः आपण लॅबोरेटरीमध्ये तयार केलेला कॅलस असेल, शास्त्रज्ञांनी केला असेल, तर त्याला शेप हा रेग्युलर नसतो, इरेग्युलर शेप आपल्याला दिसतो. तसंच आपण जर निसर्गामध्ये आपण बघितलं तरी आपल्याला झाडांच्यावरती, त्याच्या बुंध्यावरती, त्याच्या फांद्यावरती ज्यावेळेला आपल्याला ट्युमर लाईक स्ट्रक्चर, त्याला कसा रेग्युलर असा शेप नसतो, कसाही वेडावाकडा. सो देर इज अ नो रेग्युलर. इट इज डिफिकल्ट टू डिस्क्राईब द इट्स एक्सटर्नल मॉर्फॉलॉजी इन केस ऑफ कॅलस कल्चर. बट दे कॅन बी डिस्टिंग्विश्ड ऑन द बेसिस ऑफ अदर कॅरेक्टरिस्टिक्स. त्याला डिस्टिंक्शन करता येत नाही, पण दुसरे काही कॅरेक्टर्स आहेत मॉर्फॉलॉजिकली जरी आपण यूज नसलो करत, दॅट इज टेक्स्चर, कलर अँड हॉर्मोन रिक्वायरमेंट. ऑन द बेसिस ऑफ इवन callus tissue initiated from the explant of the same plant species may shows considerable variations structures are there, isn't it? अगदी एकाच प्रकारच्या एक्सप्लांटपासून जरी आपण कॅलस टिश्यू तयार केलेला असला तरी त्याच्या कॅलस टेक्स्चरच्या मध्ये आपल्याला खूप सारं व्हेरिएशन दिसेल. ओके, ऑन द बेसिस ऑफ टेक्स्चर कॅलस आर ऑफ टू टाईप, दॅट इज अ सॉफ्ट कॅलस अँड हार्ड कॅलस. मग अशीच सांगितलं आपल्याला. Now the difference we have to get idea about soft callus is a friable in nature. It is made up of a heterogeneous mass of cells having a minimal contact, easily can get separated, isn't it? Soft. Whenever the cells require more and more agitation for the separation from the callus, hard callus, giant callus, tracheates-like cells, packed cells, compact in nature, hard. Hard callus may be nodular in form, coloration. Hard callus color ha nodular असेल. Generally callus tissue is a creamy yellow or white in color. Sometimes it may be pigmented. Pigmentation may be uniform property of the callus. So these are the, we have to say little bit some characteristics of the hard and soft. Only two characters we have to keep in mind. Another again, it may be green in color. Sometimes white callus tissue grown under the dark condition turn into a green color after transforming into the higher conditions, isn't it? Again, some may be yellow in color, some may be yellow in color. And that color is because of synthesis of a carotenoids. In some cauliflower culture, it is the purple color due to the accumulation of anthocyanin in the vacuoles or due to the production of oxidized form of a dihydroxyphenylalanic. So that color depends upon which type of changes have been taken place into that one, which type of pigmentation has been takes place into the callus. The principal procedure of callus formation that is well known to you: aseptic preparation of a plant material, selection of a suitable nutrient medium, incubation of a culture under the controlled physical condition. These are the principles, and we have to say that the procedure. What is the significance of callus culture? Most important is that why we have to run behind the learning of this technique, why we have to produce the callus, and what is the exact usefulness to the human being or to the scientist, to the nature. Callus culture has a no major importance unless and until it is for other experimental objectives. Unless and until it is used for other experimental objectives. म्हणजे काय? What is things are there? We know that ज्वारी. Simple example I will tell to you. ज्वारी आहे. ज्वारी आपल्याकडे आहे म्हणजे संपलं का? आपण ज्वारी कच्ची खाणार आहे का? नाही. तर त्याचं काय करणार प्रॉडक्ट पुढचा? तर त्याचं पीठ करावं लागेल. पिठापासून पुन्हा भाकरी नंतर आपण खातो. पण डायरेक्ट तुम्ही भाकरी मागितली तर नॉट पॉसिबल. सिमिलर कंडिशन त्याचा पुढं यूज केला जातो. ज्वारीचा पुढं यूज, आधी ज्वारी तयार केली, पिकवली आणि मग त्याचे प्रॉडक्ट तयार केले. ज्वारीच तयार केली नाही तर प्रॉडक्ट तयार होणार. सिमिलर वे कॅलस जर तयार झालं तर पुढच्या गोष्टी म्हणजे कॅलस डायरेक्ट युजफुल नसला तरी त्याचे पुढचे काही गोष्टीसाठी कॅलसची गरज आहे. बेसिक मटेरियल आहे कॅलस म्हणजे. द होल प्लांट कॅन बी रिजनरेटेड इन लार्ज नंबर फ्रॉम द कॅलस टिश्यू. आपण प्लांट तयार करू शकतो. इजंट इट? अनदर थिंग इज दॅट मॅनिप्युलेशन ऑफ न्यूट्रिएंट अँड हार्मोनल कॉन्स्टिट्यूशन इन ए कल्चर मीडियम. जर कॅलस कल्चरमध्ये काही हार्मोनल आणि हे न्यूट्रिएंट मीडियममध्ये चेंजेस केले तर डायरेक्ट त्याच्यापासून आपल्याला प्लांटसुद्धा तयार करता येतात. दिस फेनॉमेनॉन इज नोन ॲज प्लांट रिजनरेशन ऑर ऑर्गॅनोजेनेसिस ऑर मॉर्फोजेनेसिस. म्हणजे कॅलस तयार झाला तर व्हॉट इज द फर्दर. आत्ता आपण काय स्टडी करतो? वी आर लर्निंग अबाउट द ओनली कॅलस कल्चर. कॅलस कल्चर स्टडी केला, स्टॉप झाला. पण सिग्निफिकन्स त्याचे फायदे बघायचे असतात, पुढे जाऊन त्याचे प्रॉडक्ट आहेत, पुढे जाऊन त्याच्यापासून काय होतं. तर कॅलस कल्चर समजा आपण कॅलस घेतलं, जर पुन्हा असं न्यूट्रिशन मीडियम तयार केलं, त्याच्यामध्ये चेंजेस केले तर व्हॉट हॅपन्स ॲक्च्युली मॉर्फोजेनेसिस टेक्स प्लेस, ऑर्गॅनोजेनेसिस टेक्स प्लेस अँड डायरेक्टली वी कॅन प्रोड्यूस ए सीडलिंग्स, we can produce some small plants from this callus. Callus tissue is a good source of genetic variability. My friends, keep in mind, it is a good source of a genetic variability is there. सेल सस्पेन्शन कल्चरची मूविंग लिक्विड मिडियम कॅन बी इनिशिएटेड फ्रॉम द कॅलस कल्चर. म्हणजे समजा तुम्हाला सांगितलं, माझ्या मगाशी मी तुम्हाला एक्झाम्पल दिलं, भाकरी करा. तुम्ही काय करणार? तुमच्याकडे ज्वारी पाहिजे, ज्वारी दळून त्याचं पीठ पाहिजे, तर भाकरी होणार. तसं तुम्हाला सांगितलं सस्पेन्शन कल्चर तयार करा. डायरेक्ट सस्पेन्शन कल्चर तयार करता येत नाही. डिरेक्टली इट इज नॉट पॉसिबल बिकॉज यू हॅव टू फर्स्ट ऑफ ऑल फॉलो द मेथड दॅट इज ऑफ ए फॉर्मेशन ऑर प्रोडक्शन ऑफ ए कॅलस and then you will have to go for the suspension culture. So suspension culture can be initiated from the callus culture. Callus culture is very useful to obtain the commercially important secondary metabolites, my friends. Secondary metabolites having a lot of importance in the life of the human being in case of the medicines, and that secondary metabolites can be produced by using callus culture technique. Several biochemical assays can be performed on the callus. Again, after significance, we have to get idea about applications of callus. Callus culture of a slow growth plant tissue system in a static medium. This enables to conduct several studies related to many aspects of the plants are given below. My friends, कॅलस कल्चर ज्या वनस्पतीमध्ये अतिशय स्लो प्रोसेसनं तयार होतं, त्याच्यामध्ये अनेक गोष्टींचा अभ्यास करता येऊ शकतो. मेनी थिंग्स वी कॅन स्टडी अबाउट द प्लांट अँड देन वी विल बी एबल टू आर्टिफिशियली दॅट प्लांट ग्रो ॲज पर our need as many as possible plants we can develop. For example, suppose we come to know that the nutritional requirement of the plant because of study of a callus culture of that plant. त्याचा कॅलस कल्चरचा स्टडी करत असताना आपल्या लक्षात आलं की कुठली त्याला न्यूट्रिएंट्स पाहिजेत, कुठल्या न्यूट्रिएंट दिल्यानंतर त्याची वाढ चांगली होते, हेल्दी होते, त्याला चांगल्या पद्धतीनं लवकरात लवकर फळं येऊ शकतात, याचा अभ्यास आपण कॅलस कल्चरवर करू शकतो. सो न्यूट्रिशन रिक्वायरमेंट ऑफ द प्लांट कॅन बी स्टडीड ॲट द टाईम ऑफ कॅलस कल्चर ऑफ दॅट प्लांट. अगेन सेल अँड ऑर्गन डिफरन्सिएशन कसं होतं, हाऊ इट टेक्स प्लेस, that also we can study with the help of callus culture. That is also application of a callus culture. Development of suspension and protoplast culture. आपल्याला प्रोटोप्लास्ट कल्चर जरी तयार करायचं असेल आणि सस्पेन्शन कल्चर तयार करायचं असेल तरी आपल्याला कॅलस कल्चर तयार केल्यानंतरच करता येऊ शकतो. सो कॅलस कल्चर हॅविंग दॅट इज इम्पॉर्टन्स. सोमॅक्लोनल व्हेरिएशन, जेनेटिक ट्रान्सफॉर्मेशन, प्रोडक्शन ऑफ सेकंडरी मेटाबॉलिट्स अँड देअर रेग्युलेशन, या सगळ्याचा अभ्यास एव्हरीथिंग इज पॉसिबल टू अस ओनली व्हेन अँड व्हेन we will have idea about callus culture. So these are the applications of a callus culture are there. With these, my friends, I will say to you, thank you very much. Again, I will tell to you, callus is an unorganized, undifferentiated and undifferentiated mass of a tissue is there, and that we can produce by using a explant, sterilized plant, small part of a leaf, root, leaf or flowers or shoot of the stem, tips of the stem. and simple methodology is there. In case of production of a callus is there, and without callus formation, without learning technique of a callus production, we will not go for the suspension culture, we cannot go for the plastic culture. Again, lot of importance of this learning of a callus culture technology there. With this, thank you.